In 2016, the Nobel Prize in Physiology or Medicine was awarded for the discovery of a biological process that every human body can activate for free, requires no equipment, no supplements, and no special food. The process is called autophagy. The word comes from the Greek for self-eating, and what it describes is one of the most elegant maintenance systems in biology, the mechanism by which cells identify damaged components, dysfunctional proteins, and cellular debris, break them down, and recycle the parts to build new functional structures. Yoshinori Ohsumi won the Nobel Prize for mapping the molecular machinery of this process. His work established that autophagy is not a passive background process. It is an active, regulated system that the cell ramps up under specific conditions and that has measurable effects on cellular health, inflammation, aging, and disease resistance. The condition that most reliably activates autophagy is one that most people in the modern world almost never experience: not eating for an extended period. Specifically, the research suggests that meaningful autophagy induction begins at around 16 hours of fasting in humans. Not days. Not extreme caloric restriction. 16 hours, which for most people means finishing dinner at 8 in the evening and not eating until noon the next day. This is not a diet in the conventional sense. It is not about what you eat. It is about the metabolic state your body enters when it has been without food long enough that several interconnected biological systems shift simultaneously. Understanding what those systems are and what they do changes how most people think about meal timing, not as a strategy for eating less, but as a way of accessing a biological maintenance mode that the body was designed to use regularly, and that most modern eating patterns prevent from ever activating. The first thing that happens when you stop eating is straightforward. Blood glucose drops. This is normal. The elevated glucose from your last meal is absorbed, used, and cleared from the bloodstream over several hours. In response to falling blood glucose, insulin levels decline. Insulin is the hormone that signals the fed state. Its presence tells cells that glucose is available and directs the body toward energy storage and anabolic processes. When insulin falls, this signal disappears. As insulin continues to decline over the first several hours of fasting, a second hormone rises, glucagon. Glucagon signals the opposite state, energy scarcity, and directs the liver to release stored glucose into the bloodstream to maintain blood sugar levels. This glucose comes from glycogen, the form in which glucose is stored in the liver and muscles. Glycogen stores are finite. Under normal conditions, liver glycogen is depleted within approximately 12 to 16 hours of fasting. As glycogen runs low, the body begins shifting its fuel production toward a different pathway. Fat cells begin releasing fatty acids into the bloodstream. The liver converts these fatty acids into molecules called ketone bodies: beta-hydroxybutyrate, acetoacetate, and acetone. Ketones are an alternative fuel source that most tissues, including the brain, can use in place of glucose. This transition from glucose-based metabolism to fat and ketone-based metabolism is what researchers call the metabolic switch. It takes place gradually, becoming significant somewhere between 12 and 18 hours of fasting in most people, and it is associated with a set of physiological changes that go well beyond simply burning fat. The brain's response to ketones is one of the more consistently reported subjective experiences of extended fasting, and the biology behind it is worth understanding. Glucose metabolism in the brain produces reactive oxygen species as a byproduct, the same free radicals discussed in the context of oxidative stress. Ketone metabolism is significantly cleaner. It produces fewer reactive oxygen species per unit of energy generated, reducing the oxidative load on neural tissue. Ketones also appear to have direct effects on brain function beyond simply serving as an energy source. Beta-hydroxybutyrate, the primary ketone body, activates a signaling pathway that increases the production of BDNF, brain-derived neurotrophic factor, the protein that supports the growth and survival of neurons and the formation of new synaptic connections. BDNF is sometimes described as fertilizer for the brain. Higher BDNF levels are associated with improved learning, better memory consolidation, greater cognitive flexibility, and reduced risk of neurodegenerative disease. The relationship between fasting, ketone production, and BDNF elevation is one of the mechanisms through which caloric restriction and intermittent fasting have been associated with cognitive benefits in animal models and preliminary human research. The subjective experience that many people report during extended fasting, a clarity of thought, a reduction in mental fog, a sharpening of focus, is consistent with what the biology would predict. It is not guaranteed, and it varies significantly between individuals and depending on how adapted to fasting someone is, but it is not coincidence and it is not placebo. Now, to autophagy specifically, the process that earned the Nobel Prize and represents the most significant cellular maintenance effect of extended fasting. Under normal fed conditions, the cell's resources are directed toward growth, reproduction, and activity. Autophagy, the process of identifying and recycling damaged cellular components, runs at a low baseline level. There is no strong pressure to prioritize maintenance when the environment is abundant. When the cell enters an energy-scarce state, signaled by falling insulin, falling glucose, and falling activity of a nutrient-sensing protein called mTOR, the autophagy machinery ramps up significantly. The cell begins actively tagging damaged proteins, dysfunctional mitochondria, and cellular debris for degradation. These tagged components are enclosed in a membrane structure called an autophagosome, which fuses with a lysosome, a cellular compartment containing digestive enzymes, and the contents are broken down into their component parts. Those parts are not wasted, they are recycled, used to build new, functional proteins and cellular structures. The process is simultaneously a cleaning mechanism and a resource recovery system. The consequences of autophagy being activated regularly are significant across multiple organ systems. In the brain, autophagy clears the aggregated proteins, including misfolded tau and amyloid beta, that accumulate with age and are associated with neurodegenerative diseases, including Alzheimer's and Parkinson's. Dysfunctional neurons that would otherwise degrade brain performance are broken down and recycled. In the immune system, autophagy is involved in the clearance of intracellular pathogens, bacteria and viruses that have managed to enter cells. It also regulates the life cycle of immune cells themselves, removing aged and dysfunctional immune cells and supporting the production of new ones. In metabolic tissue, liver, muscle, fat, autophagy clears dysfunctional mitochondria, the cellular structures responsible for energy production. Mitochondria accumulate damage over time, and dysfunctional mitochondria produce more reactive oxygen species and less ATP than healthy ones. Replacing them through autophagy-mediated degradation and mitochondrial biogenesis improves the efficiency of cellular energy production. This is part of why people who practice intermittent fasting consistently report improvements in energy levels over time, not the immediate energy of a stimulant, but a more stable, sustained energy that reflects improved mitochondrial function. Inflammation is a central mechanism in the aging process, and in most chronic diseases, and fasting has measurable effects on inflammatory markers that are worth understanding. During the fed state, particularly after meals high in refined carbohydrates and processed fats, the immune system receives signals that promote inflammatory activity. Insulin itself has mild pro-inflammatory effects at the concentrations produced after high glycemic meals. Products of advanced glycation. Compounds formed when glucose reacts with proteins accumulate in tissues and drive chronic low-grade inflammation. During fasting, insulin falls, glycation products are not being generated, and the metabolic state shifts in a direction that is consistently associated with reduced inflammatory signaling. Beta-hydroxybutyrate, the primary ketone body, has been shown to directly inhibit the NLRP3 inflammasome, a protein complex that drives the production of pro-inflammatory cytokines, including interleukin-1 beta and interleukin-18. A study published in Cell Metabolism in 2019 followed participants through a 5-day fasting mimicking diet and found significant reductions in multiple inflammatory biomarkers, with some effects persisting for months after the fasting period ended. The researchers attributed this to a combination of autophagy activation, stem cell regeneration in immune tissue, and direct anti-inflammatory effects of ketone bodies. This anti-inflammatory effect is one of the proposed mechanisms behind the associations between intermittent fasting and reduced risk of cardiovascular disease, type 2 diabetes, and certain cancers that have been observed in epidemiological research. The evidence base here is not yet at the level of established clinical recommendation. The research is ongoing, but the mechanistic picture is coherent and consistent with multiple lines of evidence. Here is the practical picture of what a 16-hour fast actually involves for most people. If dinner ends at 8 in the evening and the first meal of the next day is at noon, the fasting window is 16 hours. 8 of those hours are sleep. The remaining 8 are the waking hours of the morning during which no food is consumed. This is the most commonly practiced form of intermittent fasting, sometimes called the 16-8 protocol. It does not require skipping meals in any dramatic sense. It requires moving breakfast to later in the day. During the fasting window, water, black coffee, and plain tea do not meaningfully interrupt the fasted state. They do not raise insulin or glucose significantly and do not appear to impair autophagy induction. Adding sugar, milk, or cream does break the fast by raising insulin. The bawam say bam bawam say dam deso ben dem depends on the amount added. The transition period, the first several days to weeks of practicing a daily fasting window, is typically the most difficult. Hunger signals are partially habitual. The body has been accustomed to receiving food at certain times, and the absence of food at those times produces hunger that can feel urgent even when the body has abundant stored energy available. This hunger typically diminishes within 1 to 2 weeks as the body adapts to the fasting pattern and becomes more efficient at accessing stored fuel. The honest limitations deserve space. Intermittent fasting is not appropriate for everyone. People with a history of eating disorders should approach any structured eating pattern under professional guidance. Pregnant and breastfeeding women have distinct nutritional needs that make extended fasting inappropriate. People with type 1 diabetes or certain metabolic conditions require medical supervision before modifying meal timing significantly. And for some people, the hunger and cognitive effects during the adaptation period are sufficiently disruptive to daily function that the transition requires more gradual adjustment. These are real considerations. The biology of autophagy and the metabolic switch are real regardless of them. The question for any individual is whether the benefits are accessible to them in their specific circumstances and at what pace of adoption. Here is the framing that synthesizes all of this. The body has a maintenance mode, a biological state in which it pauses growth and activity to perform repair and recycling operations at the cellular level, clearing out damaged structures, reducing inflammation, improving the efficiency of energy-producing organelles, and producing an alternative fuel that the brain handles cleanly and efficiently. This maintenance mode is not accessible during the fed state. Insulin, mTOR activation, and the abundance of available glucose signal the cell to prioritize growth and activity over maintenance. This is appropriate when food is available. Using it makes sense. But the human body evolved in an environment where food was not continuously available. Periods of fasting, not starvation, but simply the normal gaps between meals that occur when you are not eating every few hours, were a regular feature of the ancestral environment. The cellular maintenance systems that activate during fasting are not emergency responses to scarcity. They are a regular part of how the body was designed to function. Modern eating patterns, with food available at all hours, meals and snacks distributed across 16 or more waking hours, and an insulin response that never fully drops between them, keep the body in the fed state continuously. The maintenance window never fully opens. The autophagy machinery runs at its low baseline level and never significantly ramps up. Intermittent fasting is not a diet trend. It is a way of recovering access to a biological state that the body was designed to enter regularly and that the modern food environment has largely removed from daily life. The Nobel Committee gave Osumi the prize because the machinery he described is fundamental to cellular health. The fasting window that activates it is not complicated. It is 16 hours. You already sleep for 8 of them. If this changed how you think about when you eat as much as what you eat, hit the like button. Share it with someone who thinks fasting is extreme, and subscribe for more content like this. See you in the next one.